Hi, YNP. Today we have another special guest with us, Elizabeth Schneider. I'll give her the space to introduce herself to you, and there we can jump into their amazing work so far.
Yeah. Hi, everyone. It's my pleasure to be here today. So yeah, I'm Elizabeth Schneider, and I guess maybe I'll go into more detail later, but I am American. I'm from Kentucky. I did my bachelor's and master's in Kentucky, and then I was originally doing a therapy degree. And after I did my clinical psychology degree, decided I wanted to do research. So I did my PhD in the UK at University of Birmingham, and that was on eating behavior. And then I'm now doing a postdoctoral researcher position at APC Microbiome Ireland. So that is in Ireland, and a postdoc position, what we call it, is basically the in-between of having finished your PhD and establishing your own lab. So I'm in that in-between phase now. But yeah, that's pretty much me in brief. Shall I go ahead and share my slides?
Yeah, please. Go
Okay,
ahead.
great. Okay. Yeah, so today I know that the focus is kind of more on the brain and body control of appetite. So I thought I would go over a study that I did in my PhD. It's where we use intranasal insulin as a means of understanding kind of this brain and body interaction of appetite. So just a little bit of background. Insulin, I'm sure you guys are familiar with it because you might have some friends who have diabetes and have to take the drug externally. So pretty much for people with type 1 diabetes, they either don't produce any insulin or they don't produce enough. And so they'll have to give themselves injections. Or people with type 2 diabetes, which I'll go into a little bit more detail on the next slide, they have insulin, but it doesn't really work properly. So it's critical to inject insulin if you're not producing it naturally, because it is critical for survival, and without it, we would die. So insulin in the body, though, so we also call the body the periphery as well. So you might hear me say that interchangeably. But basically, you have the pancreas here underneath the stomach. And so what happens is once we consume food with a carbohydrate, so several foods have carbohydrates, really, the majority will. Basically, our body detects this high blood sugar, and as a result, insulin gets released from the pancreas, and it allows the blood sugar or the blood glucose to go into the cells, to be released and go into the cells of the tissues and muscles to be used for energy. And in that way, it's lowering the blood glucose. So it starts off high, and then it automatically goes to low thanks to insulin. And then if there's more blood glucose, it gets stored in the liver here in the form of glycogen, to where if the body detects low blood sugar, so you're kind of hungry, it's going to release this glycogen, which ultimately raises blood sugar. So it's this really critical hormone throughout the body that's constantly maintaining energy homeostasis. And people with obesity, so it can be that their body is kind of producing more insulin because maybe it's receiving too much carbohydrates through overfeeding. And so it keeps producing this excess insulin. And this is a state called hyperinsulinemia, where you've just got too much insulin. And so over time, if that state carries on long enough, and of course, this can be years, the pancreas kind of tires out and it will pretty much stop working. And at that point, that's whenever you would receive a diagnosis of type 2 diabetes. And then you would have to take insulin shots as well. So you've got this kind of in-between phase for people with obesity, where they have this kind of body insulin resistance, is what we might call it, and that hyperinsulinemia. So there's also insulin in the brain, though. And this is really exciting because it's somewhat relatively, I mean, it's been known for some time, but I think probably we don't know about it too much in society. We just think about insulin in the body for diabetes, but there is in fact insulin in the brain. And what happens is that insulin gets transported to the brain with a special kind of carrier mechanism. And it allows that insulin to be distributed all throughout the brain. And so it can go through several key areas of the brain, such as the hypothalamus, which is important for energy regulation and food intake. the striatum, which is heavily like controlled by the dopaminergic system, and it's really involved in reward and reward-based behaviors. The amygdala, which is important for mood, and the hippocampus, which is important for memory. So it's no surprise that central insulin has been found to regulate food intake, mood, and memory. And in fact, if mice get born without the receptors in the brain for insulin, they tend to overeat and will have a large body weight compared to mice with the intact receptors for insulin. And just like the peripheral insulin resistance, there's also been cases of central insulin resistance observed in obesity. So what happens here is that pretty much the central insulin receptors are no longer as sensitive to insulin. And what's really great, though, is because traditionally that would be very hard to target insulin in the brain because the brain is so hard to kind of get at. But now there is this kind of known nose to brain pathway that can be exploited through intranasal drugs. So pretty much what happens is that, and you might have taken some intranasal drugs through like sinus issues or anything like that. So you will inhale this whatever drug, and it goes straight from the nose to the brain pathway. And that's excellent because it's quite hard traditionally to get drugs to go to the brain. And obviously a lot of people want drugs to go to the brain because it's so in control of the body. But because of that blood-brain barrier, which is this the brain's kind of built-in defense barrier to stop toxins from getting in and causing infection. So it's got this really tight barrier where things can't get in. But luckily, once you give insulin through this intranasal spray, it's a small enough molecule to where it can pass the blood-brain barrier to influence the brain. And that's great because it's safe and it's very fast acting. Generally, the peak time of the drug is 20 minutes after administration. So it's quite quick and very non-invasive, because traditionally you might have to do surgery. And some of these earlier studies have been done in mice because you would have to administer it directly into the brain. So it's an exciting new avenue to kind of study insulin action more. And the reason we want to study insulin is because some of you all might know we are now living in a worldwide obesity epidemic, and the US is in kind of the top leading of obesity. So around 42% of adults have obesity, as according to the recent survey. And obesity is related to several physical health conditions such as heart disease or type 2 diabetes, as I mentioned earlier. And is also associated with a lot of mental health conditions because of the unfortunate stigma that's associated with obesity. And it's also costly for medical care. So it was costing 173 billion estimates from 2019. And people with obesity tended to have $2,000 more healthcare costs than people without obesity. So we want to do something with obesity, but unfortunately, it's quite complex, so it's not this simple, straightforward thing where it would already be solved. And that's because appetite control in general is very complicated. So we have several things influencing our appetite. It's not just one thing. And in fact, if we look here at the figure, we have this homeostatic control. So that's largely kind of governed by the hypothalamus. And that's where we have to eat to survive, basically. So if we don't eat food, we'll die. But we also have this reward system that's making food really tempting, and we want to eat it. And we particularly want to eat foods that taste really nice, that are high in energy density, so like high fat, high sugar foods. And this is being controlled a lot by the striatum that I mentioned earlier. And probably, though, what's less known is the role of cognition in appetite control. So this is mainly what I kind of focused on during my PhD. It's gaining a lot more attention, but I think we can think of it as just as an example. So if we're trying to eat healthier, we might have to tap into some of our inhibitory processes through like the prefrontal cortex to say like, oh, I actually really want to eat healthy, so I'm going to inhibit and not eat that cupcake, for example. And you also will have to tap into some of your memory, like your long-term memory, because you'll have to maybe remember that, oh, I have this goal of healthier eating, so I'm going to have to resist, the cupcake because it's not in line with the goals that I have. So cognition is also a very important mechanism behind appetite control that somewhat gets neglected in literature. And we know all of this is very important, but what we don't know is how these systems interact to influence appetite control. So this is where we cue insulin, because if you remember from the previous slide, central insulin is involved in food intake and memory and reward. So it makes it an excellent candidate to use to understand this brain and body control of appetite. So we actually use this drug, and this is called psychopharmacology, to understand what's happening. So it's just kind of like a method to understanding it. And so with this in mind, we gave 160 international units. So that is equivalent to basically 8 puffs in each nostril, so 16 total, to women with and without obesity. And we compared its effects to placebo. So we gave this as a one-off dosage. You can give it chronically, of course, but that would maybe be unnecessary in a way. It would answer different questions, but sometimes you can use an acute dosage, that one-off, to understand how things are acting. And it's a great way to, in a quicker and cheaper, because the research is quite expensive, way to understand what's happening. And so we kind of hypothesized that the intranasal insulin compared to placebo would reduce appetite and improve mood and cognitive performance. And we thought that would be for lean women and women with obesity. Okay, so we did this using a counterbalanced, randomized, double-blind, placebo-controlled crossover design, which is a big mouthful. So I'm just going to break it down in case you guys aren't familiar with some of the terms. So this is the gold standard for research design, and there's a lot of reasons. So I'll start with counterbalance. So basically, because we're giving the participants placebo and insulin, if we were to always give insulin on the first day and maybe always give placebo on the second day, well, people start like warming up, they get more comfortable, they're people at the end of the day, the participants, and they might get used to the task. And so they might actually all start performing better on the second day. And so if we were to always have placebo on the second day, you might think, oh, it's not really doing anything insulin, but it would actually be due to the order effects. So once we counterbalance it, we make it so that they're receiving half of the time insulin on the first test day and the other half on the second test day. And that's just a way to kind of eliminate these extra variables that could be affecting the results that we don't want to be affecting them. And then with randomization, because participants will come with all their individual factors, if we were to always be self-selecting, saying, oh, you're going to, receive this drug, insulin, that would bring in all these other factors that we don't want that are affecting the drug. So if we randomly allocate them, it produces a lot less bias in the data. So it's good to randomize so that the researcher doesn't know what condition they're in, and that's double blinding. So double blinding means that the researcher nor the participant knows if they're receiving insulin or placebo for that day. And that's really important because when people know that they're receiving a drug, they can act differently. And we don't want them acting any differently unless it's actually due to the drug. And then we placebo control it. And this is where it's actually a lot more complicated and more expensive. So insulin, it's not just like a tablet that you consume. It's this nasal spray, and it smells really distinct, actually. So we had to work with a pharmacy company to make a matched placebo that smelled and looked really similar to insulin. And that was actually very expensive. So that can be quite tricky, but it adds a lot more tightness into your design. So I think it's quite important to do that. And then lastly, we used a crossover design where participants received both the insulin and placebo, but on separate days. And that's really good because once you have the participants receiving both conditions, they can serve as their own control, which reduces the need for a larger sample size. So it's a lot more cost effective, and it's just a tighter design, really, because you can rule out these individual factors because we're comparing the participant to themselves on placebo. Yeah, so that's the design, and we ended up with 35 lean women, or with a mean age of around 24, and then we had 17 women with obesity with a mean age of 26.